This was watching Bridgeman-Rutledge, and this was testing done in class on aerobic exercise, which was carried out on a bike for 20 minutes. So we made different hypotheses of what we thought was going to happen. Here are examples of different hypotheses. So the null hypothesis means that there's no difference and that the observed difference is entirely due to chance or a sampling error. The single tail hypothesis is when you predict that one critical area is greater than another critical area, or predict that one critical area is less than another area. Two-tail hypothesis where the prediction is made saying the results could go either way. So we made a hypothesis for heart rate. We said that we thought that there was going to be a plateau in heart rate during the 20 minutes, and we also expect there to be an increase in heart rate in the first 3 minutes. We tested heart rate every minute using a heart rate monitor, and we also tested it before and after exercise. So for breathing rate, we also expected there to be a plateau during the 20 minutes, and we also expected there to be an increase in breathing rate within the first 3 minutes. We tested breathing rate every other minute by counting the amount of breaths the subject took in a minute when exercising, and we tested it before and after exercise. Hypothesis for energy system: we expect there to be low amounts of lactic acid present in the bloodstream between levels 2 and 4. We also expect to see high oxygen saturation of hemoglobin between 95 and 100%. We tested lactate at minutes 3, 12, and 18 using a lactate monitor. We tested oxygen saturation every 2 minutes and before and after exercise using an oxygen saturation monitor. What is steady state exercise? Steady state exercise is submaximal exercise that's performed aerobically. This exercise can continue for a long period of time but isn't working at a high intensity. When we first start aerobic exercise, there's an increase in heart rate, which will then plateau. But the increase is caused by hormones, the release of hormones such as noradrenaline, which the receptors will pick up and send to the medulla oblongata. And then the medulla oblongata then sends an impulse down the accelerator nerve to speed up heart rate, to the SA node, which will speed up heart rate. So before exercise occurs, we have an anticipatory rise. It is caused by emotions such as anxiety, which will stimulate the limbic centers within the hypothalamus, and when the release of adrenaline from the adrenaline gland will be detected by chemical neurotransmitters called noradrenaline. These receptors detect this and send impulses to the medulla oblongata in the brain, where the cardiac control center and the inspiratory control center. These will then send impulses to the heart. The cardiac control center will send impulses to the heart and the SA node, which will then cause an increase in heart rate. And the inspiratory control center will send impulses to the diaphragm and intercostal muscles to increase breathing rate. The method: we took readings of heart rate and breathing rate at rest to allow us to prove or disprove our hypothesis. We will take these readings before an anticipatory rise has occurred, so it doesn't affect the results. We measured blood pressure and oxygen saturation before exercise. After the 20 minutes, we would check heart rate, breathing rate, oxygen saturation alongside blood pressure levels again. Heart rate was measured every minute using a heart rate monitor. Breathing rate was measured every 2 minutes by counting it. Blood pressure was measured every 4 minutes using a blood pressure monitor. Oxygen saturation was measured every 2 minutes using oxygen saturation monitor. The Borg scale will be measured every 2 minutes by asking the subject to pick a number on scale. A warm-up will be completed before and after exercise to reduce the risk of injury. Heart rate: we expect the heart rate to steadily increase and then reach a plateau. Because there's enough oxygen to maintain lower intensity of exercise and supply the working muscles. Then we expect a rapid decrease after exercise has stopped to quickly return back to the normal rate. The heart rate will increase due to receptors detecting changes in the bloodstream such as adrenaline, which will then send an impulse to the medulla oblongata where the cardiac control center is located. The cardiac control center will then send impulses down the accelerator nerve to the SA node to increase heart rate. So stroke volume. Stroke volume is the volume of blood pumped out by the heart by the left ventricle per beat. Stroke volume will increase when exercise starts until it reaches a max stroke volume. There will then be a slight decrease in stroke volume, which will then plateau as there is no increased demand for oxygen as the body is working aerobically. Once the exercise is stopped, the stroke volume will decrease rapidly as there's a lack of oxygen depth to the working muscles. The average stroke volume for an average person is 70 millilitres. Stroke volume for an elite athlete is 130 millilitres, which is much higher as their hearts are more efficient and they can pump out more blood per beat. The cardiac output is the amount of blood pumped out by the heart per minute. Cardiac output will increase once exercise starts. We will then plateau as heart rate and stroke volume will both plateau. Cardiac output will plateau as there is no increased demand for oxygen once exercise is stopped. The cardiac output will rapidly decrease as there are very small oxygen depth and therefore less oxygenated blood needs to be pumped to the working muscles. The average cardiac output is 5 litres when wrestling. Elite athletes have a higher cardiac output as their hearts are much more efficient and they're able to forcefully pump more blood. So of a higher volume is able to pump out to the working muscles. When exercising, the average person will have an average cardiac output of 21 litres per minute. However, elite athletes have 35 litres per minute as their hearts are more efficient. So when we exercise, the vascular shunt occurs, which is where blood flows from areas where it's not needed to where it is needed. When we exercise, blood flows from the non-essential organs will be sent to the working muscles where it's most needed. There will be increased vasodilation of blood vessels, which will expand to allow more oxygenated blood to flow to the working muscles. There will be vasoconstriction of precapillary sphincters near the non-essential organs, which will stimulate decreased blood flow to the non-essential organs, allowing more blood to flow to the working muscles. So blood pressure. Systolic blood pressure is the amount of force exerted against the arteries immediately after systole of the left ventricle of the heart has occurred. Diastolic pressure is the force exerted against the arteries during the diastole of the heart. Systolic pressure will increase because there is increased demand for blood flow. Diastolic pressure will stay the same or slightly decrease due to increased vasodilation of the arteries, where the arteries expand, allowing for more blood to flow and release more pressure. Blood flow is the continued flow of blood around the cardiovascular system. The rate of blood flow will increase due to the increased cardiac output and stroke volume. There will be increased venous return where the blood is flowing around the body. We will then see a plateau in blood flow which will be caused by a plateau in stroke volume. Once exercise is stopped, there will be a rapid decrease in blood flow as less blood flow is needed to be pumped to the working muscles. Thermoregulation is the regulation of the body's temperature. When exercise starts, thermoregulation will increase as muscles are working harder, so there's more increased friction, which creates heat. To compensate for this increased heat, the capillaries near the surface of the skin will open up and increase in size, therefore increased blood flow. There will be increased blood flow and get rid of the heat energy. The sweat glands will also open up and the sweat will evaporate off the body and cool. The body's normal temperature is 37 degrees and it will try to maintain this. Venous return is the flow of blood back to the heart after the blood has been delivered to the different areas within the body, such as the organs and muscle tissue. We start exercise. When we start exercise, the venous return will increase and then plateau as venous return is equal to the cardiac output. After this exercise is stopped, there will be a rapid decrease in venous return as cardiac output will decrease and less blood needs to be pumped around the body. When exercising, there's increased muscular pump, which is where the muscles contract and squeeze on the veins carrying blood to the heart. The muscle contractions increase the pressure, so blood is carried back to the heart faster against gravity. This increased the blood's ability to return to the heart and puts a bigger stretch on the heart and then therefore increases stroke volume. The law states that the stroke volume of the heart increases in response to an increase in the volume of blood in the ventricles before contraction has occurred and when all other factors are constant. When there is an increase in exercise intensity, more blood flows into the heart by venous return. According to Starling's law, there will be an increase in stroke volume until it remains at a steady state, but there is no increase in exercise intensity. When exercise stops, there will be a decreased blood flow and there will be a lower volume of blood in the ventricles. There will also be a decrease in stroke volume. According to Study Mode Research website, increased volume of blood has the effect of stretching the cardiac muscle to a greater degree than normal. This stretching effect has the effect of making the heart contract much more forcibly and thereby pumping out more blood during each contraction, so stroke volume is increased during exercise. This is known as Starling's law. So energy system. The energy system is used as the aerobic glycolytic system is the main system used. However, when the first starts, the ATP system will be used for 2 to 3 seconds, and then the phosphocreatine system will be used for 8 to 10 seconds. So the stages of how the system works. The enzyme glycogen phosphorylase stimulates the conversion of glycogen to glucose, and the enzyme PFK catalyzes this. This produces pruvic acid and 2 moles of ATP. The pruvic acid reacts with the enzyme coenzyme A, which produces acetylase. This reaction allows us to access the powerhouse of the cell known as the mitochondria, which is the site of aerobic respiration. The acetyl-CoA produced from previous reaction combines with oxalic acid to form citric acid. The citric acid produces then oxidized for a cycle of reactions known as the Krebs cycle. The Krebs cycle produces carbon dioxide as a byproduct, hydrogen, 2 moles of phosphate, and oxalic acid, which allows the cycle to continue. The hydrogen atoms produced are carried through the electron transport chain along the folds of the inner membrane of the mitochondria by NAD and FAD, which are hydrogen carriers, which split into ions and electrons, and we get NADH and FADH, which take hydrogen ions and electrons through the electron transport chain. The hydrogen ions become oxidized and are removed as H2O. However, the pairs of hydrogen ions release enough energy for the synthesis of 30 moles of ATP, and in total we get 34 moles. Respiratory system. Breathing rate is the amount of breaths taken per minute. When we expect there to be an increased breathing rate, which is then plateau, as there is enough oxygen being taken in, and the intensity isn't being increased. When exercise stops, we expect to see a rapid decrease. The average breathing rate, as there is a small oxygen depth, the average breathing rate when resting is 12 breaths per minute. The body increases breathing rate due to receptors picking up and sending impulses to the brain. The average breathing rate when exercising is 30 to 4 breaths during aerobic exercise. Neuromuscular during exercise. So sensory neurons be carrying information from receptor cells to the medulla oblongata in the brain. The brain will then send impulses to motor neurons, which will create a motor neuron pathway. The motor neurons will then connect with the muscle cells to make them contract. The Golgi tendon is located with the origin and insertion of the muscle, and it detects muscle tension and then forms the brain. Muscle spindles detect changes in muscle length, and they're located in the muscle belly, and when exercising they'll be informing the brain the changes in muscle length. So tidal volume, when we first start exercising, there'll be an increase in tidal volume due to the increased resistance causing the working muscles to need more oxygen. We will then see a plateau in tidal volume as there is enough oxygen being supplied to the working muscles, and there is a maximum amount of air that our lung can fill up with. When exercise stops, there's a rapid decrease in tidal volume as less oxygen needs to be supplied to the working muscles, and the amount of air needed from one breath is decreased. The average tidal volume for an adult when resting from one breath is 500 milliliters. An elite athlete has a much larger tidal volume due to an increased lung capacity of 800 milliliters. And just to go back to the breathing rate and how it increased, the breathing rate increases due to receptors detecting changes in the bloodstream. And then an impulse is sent to the medulla oblongata, where the inspiratory control center is located. The inspiratory control center then sends an impulse down the VRG, which will cause an increase, which will cause the intercostal muscles and diaphragm to contract and increase breathing rate.